Hi, I'm Dr. John Walsh. I'm a professor in the Davis School of Gerontology and also in USC's program in neuroscience. Welcome to the laboratory today, and I want to take you for a little tour just to let you know the kind of research that we're presently doing in the lab. My research focuses on movement disorders and brain problems associated with movement disorders. And specifically, we'll take you for a little tour through the brain right now. We look at a part of the brain that modulates the movement. This is a model of the human brain, and basically the part that we work with is deep. So we'll take off the surface of the brain like so. And you see right here, this is the cerebral cortex, and it makes a connection with deep brain areas located right there. And let's see here. So this is the brain stem, and you see this ridge of material right here. Okay? And this material is referred to as the striatum, or the caudate and putamen. And what we look at is the connection of the cerebral cortex with this deep structure called the caudate and putamen. It's referred to as the cortical striatal synapse. And then we also look at the modulation from the dopaminergic center located right here, and it sends up its axons into this area here. So we have a kind of a three synapse link where we're looking at cortical to striatal, and we're looking at nigrostriatal, and then the integration of those two. Now, the type of research we do in the laboratory is electrophysiological, but we also collaborate with other laboratories here on the USC campus, as well as over at the Keck School of Medicine. Specifically, I work with Dr. Giselle Petzinger and Mike Jakowick, where we look at molecular biological changes and biochemical changes in this brain area as it relates to these movement disorders. And presently in the laboratory, we're using a number of neurotoxins that create these types of problems in this movement disorder area of the brain.
So what I do, I'll take all this brain out, all this cold solution. It's enough to be cool. Okay. So this is the whole brain. Just let me put it. Okay. And now I just have to dissect the part we need for our experiments. So this part we'll be using for experiments, and the remainder of the brain we'll save for biochemical analysis. Now the procedure should be done very precisely, because what I'm doing now, I'm just putting some glue, and I have to move fast because not allow glue to dry. Okay, I'll put some glue, and now I just take this part of the brain which I'm interested, and I have to put it onto the glue. So this is very important process here, not to drop this before it puts on. Okay, so it's glued, but of course it should be covered with a cold solution. And what I'm doing, and at the same time I'm removing this solution a bit, as if cleaning from the remainder of the glue. Okay, when I did that a couple of times, now I have to cover it with the SASF. Actually, it's a cutting solution, contains SASF and sucrose to make sodium concentration low. Okay, so now this is vibratome. I bring vibratome close to the brain, and now I just come closer to the brain with my blade. Okay. So this is first cut through the brain, and again we're somehow far from the structure, but again I don't want to go bigger steps because we can lose the structure we are interested in. So now I see that I have to go about maybe 10 revolutions down. Well, it takes some time, but it's better to make it slowly, because when you do it fast, so it damages tissue too much, and there will be less alive cells. Okay, now the time. Actually, sometimes even not necessary to touch. Okay, so it's ready. It's another 2 slices. I just position it inconvenient for me way to cut them if I have. Okay. So they separated. You see, you have to be very gentle with the slices. You shouldn't touch them. The more you touch, the more cells you lose.
Okay, hello there. This is Randy from Dr. Walsh's lab, doing some voltammetry today. What we are doing is sticking electrodes in some mouse brain slices and checking for dopamine responses. What I'm doing right now is positioning the recording electrode, which is a carbon fiber electrode, in position 4 of the mouse brain striatum. And after I do that, okay, that's in. I have to position the stimulating electrode, which is going to give a pulse of voltage that is going to stimulate the fibers in the striatum to release dopamine. And when they do, the dopamine is going to be oxidized. It is going to release its electrons, which are going to jump onto our carbon fiber electrode. And then we will see that as a change in current over on our screen here, which is what that was right there. And I'm going to save that response. All right, and now let's go see what our response looked like. I think this is it. 4-1, yes. Okay. Beautiful. This is our response. This is our oxidation peak when the electrodes jump off of dopamine onto our or when the electrons jump off of the dopamine onto our electrode. This is the reduction peak when they leave and go back to dopamine. It's lovely. It's a good response. This is a control animal, meaning it was not injected with neurotoxin. It was injected with saline solution, which is why we're getting these big, beautiful responses. If it had been injected with neurotoxin, the dopamine release would have been virtually non-existent, which is what I've been dealing with for the last week. But this is a treat because you get these really pretty responses, which you don't usually get.